Welcome to Study Loop.
Here we are looking at the differentiation of cells inside of an organism based on what type of role each cell needs to commit to in order to allow the body to function. So differentiation is where one cell will specialize into a certain type, which will be different from other cells that specialize into another type. So this could be one cell will specialize or differentiate into a muscle cell, whereas another can differentiate into a neural cell. And of course, muscle cells will carry out functions required of muscles, whereas neural cells will carry out different functions required for neurons in, say, the brain. So when we look at the DNA of both structures, these are going to contain the same accumulation of genes. So each cell is going to have the same set of DNA to work off. And these are going to include the use of two different types of genes. We have housekeeper genes, and we have genes which are more specialized. So these housekeeper genes are responsible for processes similar to every type of cell. So these will be things such as like energy production, such as through glycolysis and the citric acid cycle, or it could be through the production of certain proteins needed for, say, the cell membrane. This is different as specialized genes, which are responsible for conducting the function of each type of different cell. So these specialized genes will be different between what type of cell it is. So here, this question is specifically asking about these specialized genes and how they vary between each different cell type. So of course, when our genes have to be read, they cannot be read inside this condensed and highly formed structure. They have to be unwound so that our proteins and polymerases have access to these genetic regions that they need to transcribe and translate. So these specialized genes are going to rely on how these cells unwind their DNA. And this is going to rely on the use of histones and the modification of DNA through DNA methylation. So depending on what type of cell it is, certain histones will prevent the unwinding of certain sequences of DNA. So DNA is typically wound around histones to help condense it and keep it in an orderly form. So in some types of cells, it may never unwind because the gene never needs to be used, whereas in other cells, you may find these histones unwinding regularly. For DNA methylation, this is the addition of methyl groups onto the DNA, which is going to prevent the addition or binding of polymerase and other transcription factors. And that can prevent the gene from being read and transcribed itself. So in certain cells, DNA may be methylated in different areas where the genes are not needed. So here, the whole reliance on differentiation between cells relies on the functions and modifications of histones and DNA methylation and other certain differences between gene regulation in these different types of cells.